it's me again. I just wanted to give you a short review or overview on what's going to be on the test so that you can know what to study in case you forgot stuff. So vocabulary, you need to know the word dominant, recessive, homozygous, heterozygous, diploid, haploid, allele, independent assortment, autosome, sex chromosome, monosomy, and trisomy. I forgot one. Incomplete dominance. I'll add that. Incomplete dominance. You need to know that one too. Yeah. So that's vocabulary. For strategies, there are like a billion. Obviously, annotate. However, there are both monohybrid and dihybrid crosses on this test. Monohybrid is the little square. Dihybrid is the one with 16. Okay, I drew that extra wrong, but you know which one I mean. If you're doing a monohybrid or a dihybrid cross, you need to list the genotypes from the word problem. So if it says brown is dominant to blue, instead of just looking at that, actually write big B, brown, little b, blue. The next step is to write the genotype of the answer that you're looking for. So if it says how many of these are green and wrinkled, you need to write the letters for green and the letters for wrinkled so you can know what to look for in the box. The next thing, when you're doing a dihybrid cross, if both parents are heterozygous for both traits, then the ratio is going to be 9, 3, 3, 1. And this 1 being homozygous recessive for both traits. That will save you a lot of time when you're doing the cross. You don't even have to do the cross. So if both parents are heterozygous for both traits, the amount that will be recessive for both traits, there's only 1. So good to know. Blood typing, in case you lost your notes, here are the different blood types. For type A, type B, type O, type AB. If they mention anybody's parents having type O, then you know that they're going to be the heterozygous blood type. And then you just cross them in a normal Punnett square, depending on what the problem tells you. Sex-linked traits, which will also be on the test, quite a few times actually. The sex-linked traits here are the different genotypes. I personally don't care which trait they're talking about. I use H's for everything. So if they're talking about hemophilia or color blindness or baldness or albinism, whatever it is, just use H. So if it's a normal female, the 2 H's would be capital. A carrier would be a capital and lowercase. Affected would be 2 lowercase for female. For male, he's either normal, capital H, or affected, lowercase h. And so this is why there are more males with sex-linked diseases, usually male, because the male gets their trait from their mom's X chromosome. They don't have another X chromosome to mask this, so they just have the disorder. There are no male carriers. The next thing, pedigrees. There will be pedigrees on this test. So if it's colored in, it's affected, whether it's male or female. If it's half colored in, then that's a picture of a carrier, and usually carriers are female. If you're looking at a trait and you want to know if it's recessive, looking at the pedigree, then it'll skip a generation. So you have 2 parents that don't have it, and their child will all of a sudden have it. If it's a dominant trait, it's going to show up in every generation. Feel free to look at your notes or these letters to figure out the genotypes for the parents. There will also be karyotypes on the test. So if you want to know if a karyotype is normal, you need to look at the chromosomes and at each position count. If there's 2 chromosomes at each position, the person is normal. If there's more than 2 anywhere, then you know something is wrong. Or if there's 1 somewhere, you know something is wrong. Then you need to check the sex chromosomes. If they're female and normal, it should just be 2 X's. Or if they're male and normal, it should be an X and a Y. If there's 1 chromosome here or more than 2, that is also a problem. The last thing would be what is on the test. Ms. Erin, what's on the test? These things. So blood typing is on there 3 times. Monohybrid crosses, there's 4 of them. Dihybrid cross, there's 3. There's an incomplete dominance question. 6 sex-linked traits. 4 pedigree questions, 3 karyotype questions, and there are 30 questions total. Please study. Please, pretty please.